All right, so I'd like to start off by thanking the organizers for a really wonderful and interesting meeting. So I'm going to be talking today about learning in the brain. I am not an expert in deep learning, but I would very much like to chat with any of you who have any thoughts afterwards about potential connections between what we're finding in the brain and network models, right? So I think perhaps some of the things that we're finding might inform the construction of future network models, and maybe the network modeling that you're doing might inform future experiments that we're doing. So let me know if you want to chat. Okay, so before I dive in, I thought I'd acknowledge those who did the work. Okay, so this work was led by PhD students and postdocs at the time, Patrick Sadler and Matthew Golub, and they were co-advised by Professors Aaron Batista, Stephen Chase, and me. Okay, so when you hear the term brain-computer interface, the first thing you probably think about is a paralyzed person controlling a robotic limb with their thoughts. Okay. And while we're very interested in this as well, I think there's a use of brain-computer interfaces that has not been widely recognized that I think has as much potential, or maybe even more potential than this. And this is using a brain-computer interface to address basic scientific questions about brain function. Okay, so a central theme here in today's talk is that a brain-computer interface is a powerful tool for basic science. Okay, why might that be? Well, a brain-computer interface simplifies the brain's output interface without hopefully simplifying the complexities of brain processing that we seek to understand. Okay, what do I mean by this? Okay, so let's take a study using arm movements. Okay, so when we're studying how the brain controls arm movements, okay, the effector here is the arm. The number of non-output neurons here is all the neurons in the brain, right? Many, many millions. The number of output neurons is many thousands, right? These are the neurons that project down the spinal cord and to your limb. And typically, in any experiment, we can monitor only a small fraction of these at any given time. Furthermore, the mapping between neural activity and movement is unknown. Okay? And the effector dynamics, okay, in this case, the nonlinear dynamics of the muscles, of the various joints, those are difficult to characterize. They're possible to characterize, but because they're difficult to characterize, in most studies, one does not characterize them. Okay, and then lastly, sensory feedback. Specifically, I'm talking about proprioceptive feedback here. That type of feedback is tied to the arm. Okay. All right, so what we propose is to use a brain-computer interface to simplify matters. Okay, so in a brain-computer interface, you can record the activity of many neurons in the brain, okay? And we look at their spiking activity in this case, and we map the neural activity into movement of a cursor on a computer screen. Okay. So the mapping that we use takes on this simple form. Okay, so x is a 2x1 vector. That's the horizontal and vertical velocity of the cursor. A is a 2x2 matrix. Xt minus 1 is the velocity of the cursor at the previous time point. B is a 2 by, say, 100 vector matrix, right, if we have 100 neurons. U here is a vector. That's the activity of all the neurons that are being recorded. Okay? So if you're recording from 100 neurons at the same time, U will be a 100 by 1 vector. And then little b here is a 2 by 1 vector. Okay? So A, B, and little b are the parameters of this BCI mapping. Okay, and what the subject needs to do is to move the cursor to hit visual targets under moment-by-moment feedback, visual feedback. Okay. So now coming back to this table, okay, what is the situation that we have here with a BCI? Okay, so the effector here is the BCI cursor or a robotic limb. Okay, the number of non-output neurons is still in the many millions. It's all the neurons in the brain. Okay, but a key concept here is that for any neuron in the brain to affect the movement of the cursor, it has to send its signal through one of the neurons that we're recording. Right? Otherwise, it has no effect on the cursor. Okay. So by the way in which we've set up this BCI, we get to record from the full set of output signals that are relevant for this behavior. Okay? And this is going to be really powerful because this is going to allow us to make causal statements about how neural activity influences behavior. Okay? And by causal here, I don't mean statistical causality. I mean scientific causality, right? Whereby for any movement that we see from the cursor, we can attribute that to specific changes in the neural activity that we recorded. Okay. And also, in a BCI, the neuron to movement mapping is perfectly known, right? That's the linear equation that I showed on the previous slide that we get to define. Okay, and then the effector dynamics defined by that equation are known and can be linear, as in this case. And then the sensory feedback here, the proprioceptive feedback, is not tied to the movements of the cursor. So one could design an experiment in which one studies proprioception using this framework, although I will not be talking about that today. Yeah. Yep. Yep. So we're defining a simplified system here to try to address some questions that are currently very difficult to address using arm movements or eye movements. And I think then after we do these experiments, I think then the next step would be to bridge to arm movements and eye movements and see how the effects that we're finding here relate. Yeah. Okay. All right. So before I move on, I want to show you a video of the performance of a monkey controlling a BCI cursor using the activity recorded using one of these arrays. Okay? And the reason why I'm showing you this is because if we're going to be doing a scientific study using a BCI, we need to have good behavioral control. Okay, so I'm going to play this video. And so the monkey is controlling this green cursor to hit these blue targets. Okay? And so typical performance is about 100% correct and about 1 second movement times from the center to the target. Okay? And so by most metrics, this is about half as good as what our arms can do. Okay? But I hope that one thing you're taking away from this video is that we do have very good behavioral control. Okay? And so from this, we're set to proceed to address basic scientific questions. Yeah? Yeah, so we define the mapping, the matrices A, B, and little b, at the beginning of the experiment so that the monkey can control it very well. So after a brief training period where we have the animal imagine making movements of 2 to 3 minutes, then we can find the parameters A, B, and little b, and then we start it up, and the performance is just like this. Yeah. Was there? Okay. All right. So there are several key advantages of using a BCI over, say, arm movements or eye movements. Okay. So the first is that we get to monitor all of the neurons that directly drive movement. Okay? And this is because of the way that we set up the BCI. Right? Only the neurons that we record get to directly affect the cursor. Right? And as I mentioned, this is going to be really powerful because this is going to allow us to make scientifically causal statements between the relationship between neural activity and movement. Okay. Secondly, we can distinguish between output and non-output neurons. Okay, so you can imagine recording from 100 neurons, okay, and using 50 of them to control the cursor, and then monitoring the activity of the other 50 neurons passively. Okay, and this setup is relevant for studying how brain areas communicate with each other, because we know that if you look at one brain area, not all of its neurons project to the next brain area, right? But rather some subset of them, right? And so that could be mimicked in this sort of setup. Okay, thirdly, we can define a simple mapping, okay, like what I showed on the third slide, okay? And that allows us to test particular hypotheses very concretely, okay, as I will demonstrate in a bit. Okay, fourthly, we can change the mapping, okay, during the experiment, okay? And when we change the mapping on the subject, that induces learning. Okay, and then fifthly, we can flexibly manipulate the proprioceptive feedback, okay? And this is an advantage that I will not be talking about today. Okay, so what we're going to do moving forward is to leverage advantages 1, 3, and 4. Okay. All right. So learning, okay, requires the brain to alter the neural activity that it produces. Okay, so whether it be learning to play tennis or learning to manipulate a Rubik's Cube, something changes in your brain between when you're a novice to when you become an expert. Okay. So we're going to try to address two key questions here. Okay, the first is, why are some tasks easier to learn than others? Okay, and then secondly, how does neural activity in the brain change during learning? Okay, so let's try to address the first question. Okay, so why are some tasks easier to learn than others? Okay, perhaps it might be because that certain patterns of population activity are easier for the brain to learn to generate than others. Okay, and can we go even farther and ask, can subjects learn to produce arbitrary activity patterns? Okay, and if not, what defines the constraint on learning? Okay, so the way in which we think about the recordings is the following. Okay, so we're recording from a small number of neurons, okay, in the brain. Okay, so in our experiments, we record from about 100 neurons. Okay, here in this cartoon, I'm just showing 3. Okay, and we define a multidimensional space where each axis represents the activity level of one neuron. Okay, so we have 100 neurons. This would be a 100-dimensional space. Here, we're drawing this cartoon for 3 neurons, so we're in a 3-dimensional space. Okay, and then if we count the number of spikes produced by each neuron within a given time bin, that defines a point in this space. Okay, and if we look at many non-overlapping bins of activity, we get a scatter of points in this space. Okay, now what we can do is to define the BCI mapping in this space. Okay, so for example, we can define this BCI mapping here, where if the subject wants to move the cursor to the left, it would need to produce neural activity down here. Okay, whereas if it wants to move the cursor to the right, it would need to produce neural activity up here. Okay. So the key experimental manipulation, okay, that we used was to change the BCI mapping unannounced to the subject. Okay. So as you might expect, okay, right after we switch the BCI mapping, the subject's performance will decrease. Okay? And through trial and error, okay, the subject might be able to use this new BCI mapping again. Okay? And that takes time. Okay? And when we first did this experiment, we observed that for certain changes in the mapping, the subject, or the animal in this case, was able to learn the new mapping really well. Okay? Whereas for other changes in the mapping, the animal was not able to learn the mapping very well. Okay, and so this led us to wonder whether there was an explanation for why certain new mappings would be easier to learn than others. Okay, so our hypothesis relies on the empirical observation that the neural activity, i.e. the green points here, don't uniformly populate this entire space. Okay? Rather, many empirical studies in different brain areas have found that the neural activity can be characterized or lie close to a low-dimensional space, okay, which we call the intrinsic manifold here. Okay? And so in contrast to the manifolds that Dan Lee talked about yesterday, the manifolds that are in this work that I'll be talking about today are all linear. Okay. So, and then one thing to mention here is that this intrinsic manifold, okay, presumably reflects the underlying network constraints. Okay? So for example, consider neurons 1 and 2 here, okay, which are connected through an inhibitory connection. Okay? So you might expect the activity of these two neurons to antivary. Okay? And that's shown here, where the activity of neurons 1 and 2 antivary. Okay. Yeah? Yeah. So in what I'll be talking about today, it's going to be 45 milliseconds. All right. Okay. So here is the central hypothesis of this experiment. Okay, so we start off with this BCI mapping, okay, which the animal can use really well, okay, just like in the video that you saw. Okay, and then we're going to change the mapping in one of two ways. Okay, the first way is what we call within manifold BCI mapping, where we change the mapping, but the new mapping remains within the intrinsic manifold. The second way in which we change the mapping is what we call an outside manifold BCI mapping, where the new mapping is now outside of the intrinsic manifold. Yep? Yeah, so we defined the intrinsic manifold based on this, the activity during passive observation, when the animal is imagining making movements. Yeah. We can talk afterwards about different ways in which we can define the manifold, and I think that's an interesting topic of discussion. Yep? Yeah, great. So the animal has extensive experience with moving its arm to move a cursor on the screen to hit these targets. Okay. So because of months and months of having done a task like this, it knows that whenever it sees a cursor on the screen, it's supposed to try to push it to the target, whether it be with its arm or with its mind. Okay? And so then we go to a phase of the experiment where it's no longer allowed to move its arm. It's lightly strapped down. Okay? So it can't move its arm, but it still imagines pushing the cursor. And so what we and other labs have found is that the neural activity in motor cortex still changes in accordance with the target that's presented, even when the animal is not overtly making the arm movements. Yeah?
So is it through BCI, the before changes?
How is it defined, you mean? So at the beginning of the day, before we turn on the BCI, we have the animal passively watch the cursor moving on the screen. There is no task.
Why don't you use the BCI itself before changing?
Yeah, that's also, we could also do that. Yeah. Yeah, we'll talk about this. Okay, so our hypothesis here, okay, is that it is easier to learn within manifold BCI mappings than it is to learn outside manifold BCI mappings. Okay, so some of you might be thinking at this point, why are you even testing this? Are you wasting your time here? So let me put two things on the table. Okay, so the first thing is that a really important control for this experiment is that right after we switch the mappings, that they have equal initial difficulty. Okay? So it's not the case that right after we switch the mapping, this is really easy for the animal to use, and this is really hard for the animal to use. Okay? Rather, right after we switch the mapping, we want to make sure that the two are equally difficult. Okay? And then we watch how the animal can learn from there. Okay. The second thing to put on the table is that others that preceded us have done outside manifold mappings, even though they didn't call it that way. And what they found was that under certain circumstances, animals were able to learn to use these certain outside manifold mappings. Okay? So it's not as if these are impossible. All right. Okay. So before I show you the results, I want to give you a little more detail as to what we did, okay, to answer some of these questions that just arose. Okay. So the neural activity that we recorded on a typical day was 90 channels. Okay? And the typical thing to do, okay, in the field is to use a common filter to map the neural activity into cursor velocity. Okay, so what we did was we introduced a pre-processing step, a factor analysis. Okay, and what this allows us to do is to disambiguate between these two different types of perturbations. Okay, so what we did was we used factor analysis to identify 10 factors from the 90-dimensional activity that we recorded. Okay, and this value of 10 was determined using cross-validation. Okay, and then these 10 factors were mapped by common filter to two-dimensional cursor velocity. Okay, so in our experiments, what we had was 90, 10, 2. Okay, in the cartoons that we're looking at, we have 3, 2, 1. Okay. So the way in which we defined within manifold perturbation, okay, was to take the 10 factors and permute them before inputting them into the common filter. Okay. And so what this does is it preserves the relationship between the activity of the neurons and each of the factors. Okay. But what that changes is the relationship between each factor and cursor velocity. Okay. In contrast, for the outside manifold perturbation, okay, what we did was to permute the activity across the neurons, okay, before we input them into factor analysis. And what that does is it requires these neurons to show new patterns of covariation. Okay. Okay, so we performed Utah array recordings in the motor cortex of macaque monkeys. Okay, we used threshold crossings here, so there's no spike sorting. Spike counts were taken in non-overlapping 45 millisecond bins. Okay, and we used an 8-target center-out task. Okay, so let me show you data from a couple representative sessions. Okay, so on the left here is a representative session for within manifold perturbation. Okay, and so what's being plotted here on the vertical axis is success rate, and horizontal axis is the trial number throughout the course of one day. Okay, so this is about 1 to 2 hours, typically. Okay, so at the beginning of the experiment, we start off with this so-called intuitive mapping, which the animal can use very well, just like what you saw in the video. Okay, so we have 100% correct here. Okay, and at some time, unannounced to the animal, we perturb the mapping. Okay, and as you would expect, the performance drops. Okay, and through trial and error, the animal can learn to recover performance, and in this case, performance is recovered almost to 100%. Okay? And then at the end of the experiment, we then present the intuitive mapping again to the animal. Okay, so what we see here is substantial learning. Okay. In contrast here on the right, here is session for the outside manifold perturbations. Okay, so here what you see is that performance starts out at 100%, right? And then we introduce the perturbed mapping, performance goes down, okay? And here we do not see sort of the same recovery, right, up to 100%, but rather performance stays sort of intermediate. And then at the end of the experiment, we present the intuitive mapping again. Okay, so the main takeaway from this slide, okay, is that for the within manifold perturbations, we see lots of learning. And for the outside manifold perturbations, we see less learning. Okay, so we did this across tens of sessions across multiple animals, okay, and this effect held up, right? So the main conclusion from this experiment, yeah, this is a great point. Yeah, so no. There, across sessions, there is a distribution of initial difficulties, both here and here. Yeah? And what we had to ensure was that those two distributions were not significantly different from each other, right? And this is the important control that I mentioned earlier, which is that the initial difficulty has to be matched in the two cases. It happens not to be perfectly matched in these two sessions, but across all sessions they were. Okay, so the main finding here, okay, is that we saw greater learning for within manifold perturbations than for outside manifold perturbations. Okay, so what we see, okay, is that there is more learning within the intrinsic manifold, okay, than there is outside the intrinsic manifold. Okay, and what this implies is that the existing structure of the network can shape learning, right? Because remember that the intrinsic manifold presumably reflects the underlying network constraints. Okay. So one other sort of takeaway from this is that in systems neuroscience, dimensionality reduction is commonly used to visualize neural activity. Okay, and a question I often get is dimensionality reduction good for anything beyond visualization? Okay, and I think the answer, this is an example, okay, where dimensionality reduction was used as part of the closed loop experiment, okay, and in this case to reveal causal constraints on what a population of neurons is able to produce in its activity. Yeah? Yeah. So we made that comparison, and there was no detectable difference in the manifold before and after learning. Okay, and so what we did was we did a simulation with realistic levels of noise to ask how large would the change in the intrinsic manifold have to be for us to be able to detect it. And it turns out that the change would have to be rather substantial. So yeah, so what we find here, right, so the data all support this idea that within the time course of 1 to 2 hours, which was this experiment, that it is difficult for the neural activity to leave the intrinsic manifold. Yeah. Yep.
What extent does the manifold, the structure of the intrinsic manifold is dictated by the activity or the area of the brain that you're recording? I mean, you can imagine, for example, there is like a
Yeah, so are you saying like maybe we could do this experiment in a different brain area? What would it look like? Or
even if the activity in one of these electrodes is really like very, very weak or there is hardly any activity there, then it's very, you don't really have the 90 dimension, the theoretical 90 dimension. In some of the dimensionality is reduced because of mechanics and not because the structure of the reason the true structure of the
yeah, so in that case, okay, so what the intrinsic manifold is trying to capture is shared co-fluctuations, right, among the neurons in their activity, right? So you're absolutely right that if a neuron does not change its activity, right, then it would not be part of the intrinsic manifold. But that's okay, right? Because still, I think then the intrinsic manifold is trying to describe the covariation among the neurons that are changing their activity. So, yeah.
But then in that case, we outside, we're trying to force the multiplicity outside of this manifold is just impossible because of chemical.
Oh, okay, okay. So an important thing that we did in this experiment, okay, which I haven't mentioned, okay, is that before passing the neural activity into that chain, we z-scored the activity of every neuron, okay? And so that is to try to protect against this, this possibility where some neurons have a higher dynamic range of firing rates and others have lower dynamic range. Yeah, so importantly, in these experiments, we are not trying to get neurons to fire outside of their natural dynamic range. Yep.
There could be cases where then usually assign dimensions, and in other cases where dimensions are extremely unbalanced. So if you
Yeah.
see anything like that, then we'll talk about it.
Yeah, typically what we see is an eigenspectrum that drops pretty sharply. I've never seen an eigenspectrum that's pretty flat. Yeah, okay. How much time do I have? Yeah, how much time? Oh, okay, well, great, I will continue. Okay, so, okay, thanks. So the second question, okay, that we want to ask, okay, is how does neural population activity change during learning? Okay, so what we're going to do is to focus on the within manifold perturbation sessions because those are the ones where we saw the most learning. Okay, and then what we're going to do is to now look down into the intrinsic manifold and ask how is the neural activity reorganizing during learning within the intrinsic manifold. Okay, so before learning, we have a picture like this, okay, at the beginning, where the intuitive BCI mapping might be like this, so that if the animal wants to move the cursor to the right, it produces neural activity here, and if the animal wants to move the cursor to the left, it produces neural activity here, right? And then we switch the mapping, okay, unannounced to the animal, okay, and now the animal needs to learn to use this new mapping. All right. So the key question that we asked here was, how does this neural activity reorganize during learning of this mapping? Okay. All right, so our approach is the following. Okay, so we formed 3 different hypotheses, okay, about how the neural activity reorganizes during learning. Okay. Then what we did was, according to each hypothesis, we predicted what the post-learning neural activity would look like. Okay? And then we compared those predictions with the data, okay, to see which of the 3 hypotheses best agreed with the data. Okay, so here's hypothesis 1. Okay, so it's called realignment, okay, and you can think about this as maximizing behavioral performance. Okay, so what happens here is that, remember that the clouds originally were here, right? And what they did was that they migrated, right? So now, when the animal is intending to move to the left, the neural activity is here, and if the animal is intending to move to the right, the neural activity is here. Okay, so a strategy like this would maximize the animal's performance using this perturbed mapping. Okay, and I should say that when we started this study, I would have put a lot of money on this one winning. Okay. Hypothesis 2 is called rescaling. Okay, and you can think about this as restoring the influence of neural activity on cursor movements. Okay, and this is in analogy to visual motor gain adaptation, okay, whereby, in this case, changes in neural activity horizontally used to have a pretty big effect on the cursor when the mapping was like this. Okay, now, with this new mapping, when the neural activity moves in this direction, its effect on this mapping is attenuated. Okay, so according to this hypothesis, the variance of the neural activity scales in a way to compensate for that. Okay? In this case, it scales horizontally, okay, to restore the variance when projected onto this axis. Okay. Hypothesis 3 is called reassociation. Okay? And you can think about this as repurposing or recycling existing activity patterns. Okay. So what I've done here is to take the original clouds and just recolor the points. Okay? Where the points are colored so that the green ones are all more towards the left, and then the purple ones are all more towards the right. Okay. So then we set about trying to test which of these 3 hypotheses best agree with the data. Okay, so I should mention, okay, that all of these hypotheses predict after learning neural activity that is within the intrinsic manifold, okay, that respects the physiological constraints on the firing rates of individual neurons, okay, and also it has realistic neural variability for each movement direction. Okay, so the first thing we did was to look at the neural activity, right, to visualize, to see which of the 3 hypotheses would be most likely. And what we saw were pictures like this, where the cloud, the overall cloud of neural activity before learning and after learning were extremely similar. Okay, we took almost any two-dimensional projection of the neural activity, and we would get a picture like this. Okay, that was the first clue that something interesting might be going on here. The overall cloud does not seem to be changing much. Okay, so then what we did was to try to quantify this. Okay, and so we defined a repertoire change metric, okay, whereby 0 here means the cloud stays rock solid. Okay? A positive value here means that the cloud either shifts or expands. And a negative value means that the cloud contracts. Okay. So according to each of the 3 hypotheses, okay, they would predict the following. Okay? Realignment and rescaling would both predict a positive repertoire change. Okay? Whereas reassociation, as you would expect, would predict 0 repertoire change, right, based on the cartoon. Okay, so what did we see with the data? We saw this. Okay? So the data, okay, showed no repertoire change, consistent with reassociation. Okay. Another metric that we used was a change in variance, right? What is the variance of the cloud along different directions? Okay, the horizontal axis here is the variance, okay, along the intuitive mapping. The vertical axis here is a variance along the perturbed mapping. Okay. So according to hypothesis 1, what learning does, or post-learning, we see an increased variance along the perturbed mapping. Right? So hypothesis 1 would predict points up here. Okay? And these 3 correspond to 3 different animals that we had. Okay. In contrast, hypothesis 2 would predict an increase in variance along the intuitive mapping, right? And so the prediction for rescaling is here. Okay. And then reassociation predicts no change in variance, right, along either axis. Okay? And so reassociation, the blue points, predict here. Okay? Now when we compare to our data, okay, that's the black here, we see that the data show almost no change in variance along either axis, consistent with reassociation. All right. So in addition to looking at the neural activity, or quantifying how the cloud of neural activity changes, we also asked how well can each of the 3 hypotheses predict the behavioral learning that we observed during the experiment. Okay, so a typical effect during an experiment is that when we look at the animal's acquisition time, right, this is the time it takes to move the cursor from the center to the target, okay, starts off at about 1 second. When we change the mapping, the acquisition time, as you would expect, goes up. Okay? And then during learning, the animal can decrease its acquisition time, but typically this acquisition time does not come back to its original level. Okay? So another way of saying this is that learning over the course of 1 to 2 hours here is suboptimal. Okay? We have not seen a case where on this time scale, the curve comes all the way down. Okay. Now we can ask, what does each of the 3 hypotheses predict? Okay. So what we can do is to take the neural activity that's predicted by each of the 3 hypotheses, pass them into the perturbed mapping, and that gives us a predicted performance level after learning. Okay? And what we see here is that reassociation does a good job of predicting this suboptimal learning. Okay? Whereas the other 2 hypotheses predict more learning than what the animal actually showed. Okay, so all of these different lines of evidence suggest, okay, that this hypothesis is consistent with the data, okay, whereby during learning, okay, neural activity is difficult for the neural activity to move outside of this so-called neural repertoire. Okay? Instead, what it seems like is happening is that the animal can choose different activity patterns within the repertoire, right, to improve behavioral performance. Okay, so I'm just going to sum up. Okay, so in part 1, okay, what we found was that learning was constrained by an intrinsic manifold. Okay, and now in part 2, what we're finding is that within the manifold, there are further constraints. Okay, and this constraint is described by this neural repertoire. Okay, and as I mentioned, what we found was that learning is suboptimal on the time scale of 1 to 2 hours. Okay, and then we have ongoing experiments where instead of having the animal learn during 1 or 2 hours, the animal learns over 2 weeks. Okay, and over 2 weeks, what we find is that it appears that both of these constraints can be violated. Okay, and so what this implies is that these constraints can be violated, but it just takes more time. Okay, and now I'm going to skip to the very last slide because I want to connect, right, to deep learning and network models and pose a couple questions that I would be very happy to discuss with you afterwards. So what we're finding with the neural repertoire perhaps is a constraint in the brain that provides a balance between flexibility and stability in learning. Okay. And a question is this a way, perhaps, by which the brain avoids catastrophic forgetting, right, which we often see in network models? And furthermore, how do these network constraints give rise to the neural repertoire? Right? So everything that I've shown so far are empirical observations, right? And I think it would be very interesting to construct a network model that has the constraints that we're observing. Okay, that's it. Thanks.